Hello, my name is Charnelle Wickliff. I am a graduate student at Moss Landing Marine Labs, and I am a Wrigley Summer Fellow. My presentation will be on my work as investigating the effectiveness of aerial monitoring of spatial and temporal changes of Santa Catalina Island rhodolith beds. Catalina Island is teeming with life, both in land and sea. It's home to foxes, bison, deer, as well as spiny lobsters, Garibaldi, and many algae species. One algae species are called rhodoliths. Rhodoliths are calcifying coralline algae that creates beds of spherical, unattached, branching individuals that roll around on the seafloor. Many have called them the tumbleweeds of the sea. This habitat creates a dynamic 3D habitat, supports a diverse community of juvenile inverts, fish, and other algae species. In many places, they provide a stable habitat for juvenile commercial fisheries like clams, pinshells, and corals. Rhodolith beds in Santa Catalina may be important nursery ground for juvenile species that feed spiny lobsters, an important fishery species. There are 7 distinct beds along Catalina in 6 coves. There's Emerald Bay, there's Cherry Cove, 4th of July Cove, Isthmus Harbor, Avalon Harbor, and Big Fisherman Cove. The depths of these beds range from about 40 meters to 21 meters with an average of 10 meters or 30 feet, according to Tompkins and Stellers in 2016. Little is known about the temporal variation in beds cover and shape. Distribution and abundance can be influenced by physical and anthropological factors. Rhodolith beds typically occur in areas of moderate water motion where the movement is necessary for the beds to roll around and prevent bile falling and sediment accumulation. Some physical influences that can change and affect a bed are seasonal changes, winds and currents, and winter storms. Some anthropogenic factors are mooring chains, especially in a paper they mentioned with a boat on mooring chains. It was in Tompkins' 2001 paper. The boat on a mooring ball can actually make the mooring scarf in the chains even more visible and exacerbated. And then there's bioturbation, where stingrays and fish are burrowing and moving around the rhodoliths to get to food sources. So there are different methods that exist in monitoring the rhodolith beds. Satellites have been used to monitor the shape of the beds and other algae species. With satellites, it is difficult to find good resolution, finer than 1 meter resolution. Most free satellite data can miss the finer details within smaller habitats. Diving is a great way of looking at small habitats in great detail, but is limited to bottom time. Diving is also labor intensive in suiting up and getting regulators and all the equipment needed just to dive alone. And then you have to add on the surveying equipment that is necessary before you can actually go into the water to monitor these changes. Technology now exists that may address the issues between diving and satellite imagery. Drones can cover large habitats in less time than diving, and have a finer scope of resolution than satellite images. Equipped with high-resolution camera and polarized lenses, drones are able to capture a complexity and composition of a habitat within a few centimeters of resolution scale, filling in the gaps between both diving and satellite images. For the purpose of my research, I will be comparing dive data with drone data, and my analysis determine if drone data is accurate and can be used in place of diving. So my questions are, can drone imagery and diver survey detect spatial and temporal changes and distribution features, perimeter, which is the edge of the bed, and cover, which is both the area within the perimeter and the percentage of live cover of rhodoliths of Santa Catalina Island's rhodolith beds? The second question is, can scars from the crushing disturbance of mooring chains in Catalina Island rhodolith beds be detected from drone imagery? So before I started my thesis project, I had to choose the locations. So the purpose of my surveying, I chose Emerald Bay and Isthmus Harbor. They are similar in depth and have the highest percent cover of live rhodolith forming the beds. Both are on the northeast side of the respective cove, with Lion Head Point protecting Emerald Bay. The two beds have extensive mooring damage and have been surveyed for rhodolith cover in 2018 and 2019 by Stellar and Edwards, a part of a Sea Grant funding. The second thing is I had to test the drone's ability to actually capture the rhodolith beds. In July 2019, I flew the drone over both Emerald Bay and Isthmus at 3 different heights, 60 meters, 80 meters, and 100 meters high, with varying image overlapping, meaning when the drone took a picture, it adjusted how much that picture overlapped with the neighboring photos. And those were 85, 90, and 95 overlapping. I also went out and flew the drone at 2 different time periods, one in the early morning where there's less wind and more cloud cover. The next time was later in the afternoon, the sun was out, and there wasn't a whole lot of cloud covering. And also at different times of tide, one with low tide and one with a higher tide. We found that lower tide is better just because I fly from the actual beach itself. The other things that we found out from the preliminary study was that 95% image overlap was best to actually create an image, and also 80 meters was best to actually be able to capture as much of the rhodolith bed as it flow over as possible. The image that you see is actually from that preliminary study. This is Isthmus Harbor, and it shows here, from here on this way, those are rhodoliths. This is actually eelgrass, and that was ground truth or determined to be true by divers who dive this area as well. So to address my first question, can drone imagery and dive survey detect spatial and temporal changes in distribution features, of perimeter and cover of the Santa Catalina Island rhodolith beds? So to determine if they both can detect it, divers will be swimming the perimeter of the beds with a buoy. This buoy will mark their location as they swim the edge, which is where the live rhodolith, within about 0.5 meters of the edge or live rhodolith cover. They swim the perimeter with a buoy, and there's a boat driver and a person on the boat with the GPS location. When the divers are at the perimeter of the bed, they will pull the buoy about 3 times, and that's where the person on the boat will take a GPS point, and they will follow the divers around the perimeter of the bed until they've completed the perimeter. Once that is done, the person on the boat will take the buoy from the divers, and the divers will then go to the center of the rhodolith bed and take photos of a quadrant that will estimate the percent cover of the bed along a 20-meter transect. The drone will fly over these beds as well and will then be incorporated in ArcGIS with the GPS points, so that then the drone imagery and the dive data can be compared as to what the drone sees as the perimeter, captures as the perimeter, and what the GPS points from the divers actually collect. Similar with mooring chains, divers will estimate the area of crushing rhodolith, the crushed rhodolith affected by the chains themselves. They will dive down and take the length of the disturbed area as well as the width of the disturbed area, and we will calculate a area of disturbance based on their information. And then with the captured image of the drone and ArcGIS, that area will be then measured in ArcGIS, and then there will be a comparison that way. So these are the initial results. On the left side is Emerald Bay, and on the right is Isthmus. As you can see in Emerald Bay, there are areas inside the rhodolith where there's sand coming through the orange circles. So this area circled in orange is the bed with a little bit of rhodolith going up this way. And along the mooring balls, you can see some of the scarring that occurs within the bed area themselves. And similar here, this again is isthmus, and along the mooring chains there, you can see some disturbances. This is just an image of isthmus with those GPS points overlaid on the image. So you can see in some areas where the diving data is right next to it, and then some areas where the diving data is not. And like an image here, ArcGIS image classification may have a hard time deciding if where this data point is from divers is actually considered rhodolith in the GIS imagery. So with this, the significance of this project, is that it is a piece of a much larger research project that is being done in Catalina by Dr. Diana Steller from Moss Landing Marine Labs and Dr. Matthew Edwards from San Diego State University as a part of their Cal Grant funding. It is also to help the rhodolith community as far as providing a way that would allow researchers to survey the bed more quickly without having to actually use divers for that process. So I would just like to give a big thanks to everyone who has helped me along the way, to all my advisors, especially Diana Steller, who's been out in the field with me, Dr. Allison Hopp and Dr. Corey Garza. Without their support, I would not be able to fly and learn the drones, as well as Dr. Matthew Edwards from SDSU and the lab from out there, as well as my colleagues from Moss Landing Marine Labs. Thank you.